Now let us see about the Barr body. That is the sex chromatin. Okay, this can also be asked as a SPA. So Barr body is the inactive X chromosome seen in the interphase nucleus. So out of two X chromosomes, generally one X chromosome gets inactivated. This inactivated chromosome forms the Barr body. Okay, so this Barr body is the inactive X chromosome which is seen in the interphase nucleus. So it is present in the female somatic cells. It appears as a dense chromatin mass at nuclear periphery. It represents the condensed inactive X chromosome. Okay. So Barr body formula. There is a formula for the Barr body. Number of X chromosomes minus 1. Depending upon the number of X chromosomes minus 1, there will be number of Barr bodies. This is very, very important. This you will come to know from the examples here. Okay. Suppose we have 46 XX chromosome, that is a normal female pattern, there will be one Barr body. If it is a 46XY, that is normal female, there will not be any Barr body because there is only one X chromosome. Okay, and why the X chromosome gets inactive? Why it becomes inactive, that also we will discuss. Okay, now suppose if there is abnormal chromosome structure, that is 47XXX, then number of X chromosome minus 1. So here there are 3 X chromosome minus 1. You will have 2 Barr bodies. So when you examine the Barr bodies, you will get the idea of the karyotype. Okay. So if you have a 47XXY, which is Klinefelter syndrome, you will have one Barr body. So if you have 47XXY and there is a one Barr body, but when there is a 46XY, there is a 0 Barr body because there is only one X chromosome. Okay. If there is a Turner, okay, that is 45X chromosome, there will be again no Barr body. So here also there is a female phenotype. Here also there is a female phenotype. But if you see in normal female, there is one Barr body, while in this female phenotype, there is no Barr body. Okay. So in males, there is no Barr body. In Klinefelter, Barr body will be present even if the phenotype is male. Okay. In Turner, there will be no Barr body in spite of female phenotype. Okay. So what is the hypothesis for this Barr body formation is the Lyonization. This is proposed by the Marie Lyon. So what does this hypothesis states? It is the one X chromosome in females becomes randomly inactivated during early embryonic life. So one X chromosome out of two chromosomes will become randomly inactivated in early embryonic life. Okay, it occurs at blastocyst stage. So at which stage it is happening? It is happening very early at blastocyst stage. Inactivation is random. Any of the X chromosome, maternal or paternal, we know that when there is a XX chromosome, one is coming from the mother, one is coming from the father, and there is no specific that only the maternal X chromosome will get inactivated or only. It is random in each cell. Okay, but whatever happens in that particular cell, that is permanent in that particular cell lineage. Suppose a maternal is getting inactivated in a particular cell, it will continue in the further subsequent stages also, divisions also. Okay, so what it leads to? It leads to the mosaicism. Okay, it leads to the mosaicism. Okay, so examples of mosaicism, that is calico cats, that is different coat colors, and in humans variable expression of the X-linked disorders. Okay, so even X-linked disorders, they have variable expression, and this is mainly because of this mosaicism. Okay, so what is the mechanism of this X inactivation? We will not go into the detail because this mechanism is not asked. So there are just steps. I will read it out. One is the initiation, where X chromosome selected for inactivation. Then there is a GIST gene activation located on the X chromosome, produces GIST RNA. It codes the X chromosome, and that triggers the inactivation. And GIST RNA spreads over the entire X chromosome. Then there is a silencing. Okay, DNA becomes highly methylated, histone modification occurs, and chromosome become heterochromatin, that is inactive. So ultimately the heterochromatin, here you just remember there is a methylation. Okay, so once there is a methylation, the histones are modified, and that will lead to the heterochromatin. So this methylation might be important from the SBA point of view. Okay. So, and this inactivation is maintained in all the daughter cell lines. Okay. So initiation is X selected, then gist activation is RNA coating, then there is a spreading, covers the X chromosome, then there is a silencing, methylation happens, and maintenance, that is the stable inactivation. Okay. Now, what is the significance of the X inactivation? Why this X inactivation should happen? This you understand well. Okay. So there is a dosage compensation. This is very important. It ensures the equal gene expression in males and females, because males have XY, females have XX. So this X chromosome will carry certain information. This X will carry certain information. If both will get expressed, there will not be same expression in the males and the females. If there has to be expression in the males and females by the X chromosome, should be equal. In that particular case, one should get inactivated. It will happen only because of this Lyonization. Okay, I hope you are getting this. Okay, so it prevents the overexpression. Without inactivation, female would have double gene products. Okay, whatever is coded by X chromosome will be doubled in females, while it will be single in males. Okay, so it leads to the mosaicism, that is the variable expression of the X-linked diseases. Hemophilia carrier may have mild symptoms. Okay, then clinical relevance, like Turner syndrome, there is no Barr body. Klinefelter syndrome, Barr bodies are present. X-linked disorders, there is a variable expression. This is because of the Lyonization. Okay, so from examination point of view, remember Barr body is the inactive X chromosome. Number of Barr bodies equal to X minus 1. Lyonization is random and it happens in early embryonic life. Then GIST gene is a key regulator, leads to the mosaicism, and inactivated X leads to the heterochromatin formation. Okay, so Barr body is the inactive X, formula is X minus 1. Lyon hypothesis is the random X inactivation, and GIST leads to silencing of one X, and function is the dosage compensation.
Yes, sir.
Yeah, please
Barr
go ahead.
body represents active X chromosome, inactive X chromosome, Y chromosome, euchromatin, mitochondrial DNA. Sir, option is B, inactive X chromosome.
Yes, Barr body is the inactive X chromosome. That is the condensed inactive X chromosome seen in female cells. We have just seen that. Next.
Number of Barr bodies in a patient with karyotype 47 XXY is X minus 1, sir, 1 Barr body.
Yeah, because there are two X here. Okay, so next one.
Which of the following individuals has no Barr body? 46 XX, 47 XXX, 47. Sir, option is D, 50, 45X Turner syndrome.
Yes, very right, very right. Okay, so Turner syndrome, no extra X, so no Barr body. Next.
Lionization refers to DNA replication, X chromosome duplication, random X chromosome inactivation, gene mutation, chromosome deletion. Sir, option C, random X inactivation.
Correct, correct. So, what is this Lyon hypothesis says? Random inactivation of one X chromosome. Next.
Which gene is responsible for X chromosome inactivation? Sir, option C, GIST gene.
Yes. Option C. This gene produces RNA that codes and inactivates X chromosome. Next.
At which stage does X inactivation occur? Sir, option B, blastocyst stage.
Correct. Early embryonic life, it is the blastocyst stage. Correct. Next.
Mosaicism refers to multiple chromosomes, multiple genes, two genetically different cell population, mutation in all cells, chromosome duplication. Sir, mutation, two genetically different cell population. So mutation in all cells.
No. Mosaicism is the two genetically different cell population, because this inactivation is random. Okay. But it is permanent in that cell line. Okay. So it will lead to the different cell lines, different population. One with maternal X inactivated, one with paternal X inactivated, sort of thing. Okay, so mosaicism leads to the two genetically different cell populations. Okay, option C, mosaicism is leading to a different genetic makeup in cells of same individual.